This presentation will review DNA shearing products, describe their most popular role today, their strengths and weaknesses, and estimate the needs for tomorrow. DNA shearing is most commonly used in DNA or RNA sequencing. The first step in all DNA sequencing efforts is sample preparation. For example, cells must be processed to release their DNA, which is then submitted to sequencing. Sample preparation is therefore a very important part of the workflow. As sequencing is becoming more and more popular, particularly in the clinic, sample preparation will also become more and more important. Let's take a closer look at this workflow. Without exception, sample preparation involves the generation of two important intermediates: pure genomic or chromosomal DNA and DNA fragments. Cells are processed to obtain genomic chromosomal DNA, which is then fragmented or sheared to generate DNA fragments. The fragments are then linked to adapters, or short DNA oligos, to generate what's called the linked fragment library. This step involves the three enzymatic substeps, including a DNA polymerase, a terminal transferase, and a DNA ligase step. Next, the fragments in the linked fragment library are immobilized and amplified, generating the amplified library, which is then finally subjected to sequencing. Above each step is the average percent of time spent on workflow, calculated from a survey of about 250 scientists. As you can see, about half the time is spent in sample preparation. Unlike purification and adapter ligation steps, which are standard high efficiency and reproducible processes, the shearing or fragmentation step is temperamental and requires complicated hardware. The generation of DNA fragments is a critical step, though, as the fragments need to be of homogeneous size but randomly cut to satisfy the shotgun approach. DNA fragments can be generated with a variety of different equipment that I will briefly describe in the next slide. Here I've listed nearly all equipment currently available to shear chromosomal or genomic DNA. The top 5 are the most important and common in the marketplace and include Digilab's HydroShear, Covaris's S2 and G2, and Bioruptor's XL200 system. The HydroShear is based on mechanical or pressure stress forces and generates fragments with very good homogeneity and randomness. The Covaris instruments are based on a type of temperature-controlled sonication called adaptive focused acoustics and can generate small fragments in a random fashion, but the fragment size distribution is not tight. So which shears are the best for DNA sequencing efforts? In order to answer that question, we need to know a little more about the requirements of the sequencing step itself and the differences between the various sequencers. Here I listed all important sequencing machines. The first generation sequencer shown in the first row was from Applied Bio. The most common sequencing activities include whole genome sequencing, WGS, exome sequencing, which is the sequencing of exons, target sequencing, which is typically single genes or single nucleotide polymorphisms, transcriptome, which is essentially mRNA-derived sequencing, RNA profiling, which includes non-coding RNA sequencing, and chromatin immunoprecipitation, or CHIP, which is essentially the sequencing of DNA fragments that bind to proteins of interest. Applied Bio's machine was very good at targeted sequencing, as shown by the green highlighted color. In about 2004 to 2005 came the second or next generation sequencers, including Illumina's HiSeq 2000, LifeTech Solid, and Iron Tor systems, and Roche's 454 GSs. Unlike the first generation machines, these second gen machines sequencers can integrate fluorescent bases and detect the signal at the same time. An important question is, what is the best DNA substrate length for each of these instruments? This is predominantly determined by the read lengths of each of the instruments. While HiSeq 2000 and Solid have short read lengths of about 50 to 75 base pairs, the Iron Torrent reads about 150 base pairs, and the 454 GS reads about 400 to 500 base pairs. Typically you want your library to be about 50 to 100 base pairs larger than the read lengths. Now let's look to the future. Third generation sequencers, which are coming into the market as we speak, can read the polymerase sequencing reaction in real time and in a continuous manner, as all the nucleotides are added at the same time, and there's no need to wash between incorporated nucleotides. And there's no need for the immobilization and amplification steps either. Importantly, these instruments can sequence the human genome in about 20 hours as opposed to 200 hours, essentially an order of magnitude faster than the second generation instruments. What are the read lengths of the third generation instruments, and which shears, DNA shears, will be best for these instruments? I've listed 7 third-generation instruments, mostly still in development other than PacBio's RS system. These include systems from Oxford Nanopore, Nobelgem, Stratos, Ansys, Roche, 454 IBM, and NuBio. All of these promise to have significantly higher throughput as well as read lengths, which will all be in PacBio's range of about 1,000 base pairs, maybe in the average of 3,000 to 5,000 base pairs. Therefore, the shearers of the future will need to service this market. DNA shearers will need to generate large fragments that are very homogeneous. Let's take a closer look at HydroShear's capabilities. This instrument can be set with either a small, standard, or large ruby pore assembly. The user simply adjusts the dial, which is also known as the shear or speed code, to the value that will result in the desired average fragment size in base pairs. For example, if the user needs an average of about 2,000 base pairs, they can use the standard ruby set to shear code of about 6 or 7. If they want fragments of 12,000 average base pairs, they can set the large ruby to speed code 14-ish. The advantage of the HydroShear over other instruments is that it can generate fragments of very large size. Another advantage of the HydroShear is the tight distribution of fragment sizes. To better understand this concept, let's look at the details in the following slides. DNA fragment size is generally determined from agarose gel electrophoresis experiments. For example, shown here is the gel of purified crustacean DNA untreated and sheared by a variety of protocols. The molecular weight standards are on the left and on the right. The first lane is unsheared DNA with most of the fragments between 48 and 63,000 base pairs. The next 3 lanes are variously sheared DNA with rough averages of about 20, 34, and 48,000 base pairs. Another way to represent these results is in a graph format shown in the next slide. The image of the DNA agarose gel, shown at the top right, is scanned to measure the intensity of every pixel along a line of every lane. The pixel intensities are then plotted versus distance, which can be converted to molecular weight or base pair, in this case giving rise to the graph below. In conclusion, the HydroShear has an advantage over its competitors because it can generate the largest fragment sizes, ideal for third-generation sequencers. And equally important, generate the tightest fragment size distributions, also important for generating high-quality data. Coupled with the fact that mechanical or pressure-based shearing results in the lowest DNA damage, we believe that the HydroShear is the ideal tool for the sequencing market.